Hello. Can someone please confirm that you can hear me?
We can hear you.
Great. All right, are there any questions? If not, let's go ahead and get started. Maybe I should get the information first. Just a minute, my computer's a little slow. Trying to call up the information now. Lesson 13. All right, let me share my screen. No, I don't think I need that. Close that down. That didn't even close. All right, let me share my screen. You should be seeing my screen now. So today is August 18th, and we should be finishing chapter 13 and starting chapter 14 today. There is no lab today. I will be logging in from 7 to 7:15 to answer questions. If you do not have questions, you do not need to log into the lab. After 7:15, unless there's a bunch of questions, I'm going to log off. Obviously, I'll stay on for as long as there are questions. Any questions about what we're doing? All right, let me remind you the final is coming up. It's going to be a comprehensive final. You're going to take it on August 25th. That's a Thursday. Let me confirm that. Yeah, final is August 25th. It will be the same type of an exam as most of the quizzes, where there was multiple choice questions requiring Respondus Lockdown Browser. Most of the questions will be that way. There will be a short answer or essay questions, and that'll be in Final A. Only 15 points in Final A, 135 points in, well, I guess that's supposed to be Final B. Doesn't say B there, but it's going to be Final B. I think it's under 50 questions. Let me confirm that. I got to shut this down so you don't see the questions. This is not working. Yeah, there's stuff sharing. On the fill in the blank question, it's probably less than 5 questions, or fill in the answer. For the other multiple choice questions, I don't see it here. Let me get to the page. Come on. My computer is really working slow with Zoom running, so 35 points divided by about 3. Go to the questions because I'm not sure the number. 40. So about 50 questions. All right, any question about any of that? Let me get out of there. Start the screen, share the screen. Should be seeing my screen again. All right, any questions about what we're doing? Let me do state that the final, I thought I had that on there, but I'm not seeing it here. The final will be 50%, oops, come on. 50% of the final will be from chapters 13, 14, and lab 9, which is material you have not been quizzed on, as well as the unknown project. The rest of the questions will be throughout the term, so there will be at least one question from each chapter. A little bit more on chapter 1 because chapter 1 introduces many different topics, and then we hit them again in later chapters.
You said 13, 14, 15 is majority of it though?
No, not 15. We're not doing 15, just 13 and chapter 14. 50% of the questions will come from chapter 13, chapter 14, and the unknown project.
Okay.
Okay. The rest of the material you've been quizzed on, and so you will be tested on it, but it'll be not as many questions. All right, let me see. Is there anything else I should talk about that? The exam will cover both lecture and lab, so study both lecture and lab. There will be no questions on the infectious disease project because each person has a different project. I think that's it about for the final. I can't think of anything else to talk about. So let's go ahead and do that. The unknown project, let me remind you, you have to turn it in by Wednesday, August 24th. The unknown notes pages will close on Tuesday, August 23rd. I think I'm going to shut that down because my computer is running slowly. All right, so we were talking about viruses. Now remember we moved on to animal viruses. No, this isn't where we were. There we are. This is where we were. So we had talked about COVID-19. The symptoms of COVID-19 are mostly flu-like, though the runny nose and sneezing are less common than the flu, and the shortness of breath and the deaths are more common than the flu. Mortality rate increases with the age of the patient, with the ill health of the patient. Mortality is more likely to be seen in men, in pregnant women, and Blacks and Hispanics. Now let me state that we don't know why Blacks and Hispanics have a higher mortality rate, but this is seen more than just in the United States. That Blacks and Hispanics have a higher mortality rate. In fact, I don't know if it's still true, but the last time I looked, the country that had the highest mortality was actually Mexico. And that's not necessarily because of other reasons, although it is definitely true that they are a third world country and the disease is hitting that country more than first world countries, but it's because they are Hispanic. And for whatever reason, COVID-19 is more lethal in Blacks and Hispanics. And nobody's really understood why that's the case. In men, it's probably, and the increased risk in men is just a little bit. Blacks and Hispanics is not quite double what it is in whites and Asians, but it's close to double. So in men, it might be like 10%, Blacks and Hispanics is close to 100%. And it's just that men are weaker when it comes to an infection. So it's not surprising that it's more lethal in men. And it's because men only have one X chromosome, and having a redundant system, meaning two chromosomes, really does make a woman better at fending off diseases, better at surviving diseases, and better in many cases. Like, for example, in this country, it's well known that women live longer than men. And it's not because women don't go into the workforce and don't have the stress of that, because they certainly do go into the workforce and they have the same stress. And it's not because men are heavier smokers. Women, ever since the 1960s, have taken up smoking just as heavily as men. So it's just that men, because they only have one X chromosome, makes them at greater risk. What is a little different with COVID-19 from most other infections is that the age of the patient. Normally, let's see if I can highlight this. I'm gonna throw that down now. Normally in an infection, when we see the age of the person, let me get that on here. So we have a graph here. Oops, not a very straight line. But normally when we see illness and death from an infection, they're high at very young age, and then it decreases for older children, teenagers, young adults. And then as you get older, it starts increasing. And so we have the highest mortality rate among the elderly and among the very young. And that's very common. Okay, so most infections, this is what we see. With COVID-19, we don't really see this up here. What it is there is a slight risk at very young age. I'm talking about newborns, and then it drops very rapidly, very low. And then it increases, I don't know, around 20, age 20, and then starts increasing slowly. But it doesn't become high until about age 50. So let's put in there 50. And I don't remember if it's age 55 or age 60 that they say that there's really increased risk of death to COVID-19. Okay, and it just goes exponential. The older you are, the greater the risk of dying. Okay, and this age thing, like I said, it's really much higher than even the newborns. And then shortly after newborns, like about year one, the death drops to very low levels. So it's very slight and only really among newborns. And that's very unusual because in most diseases, it's high on the very young and then the young, and it increases with the elderly. COVID-19 just is not seen that way. And no one really knows why, just the way the disease works. they do know a little bit about why children are so good at fighting COVID-19, but I don't want to get into that right now. All right, the incubation period, you have to be infected for 2 to 14 days with COVID-19 before you'll have symptoms. Of course, some patients are asymptomatic. And if we're talking about young people like you guys and children and teenagers, actually among the young, about 50% of patients are asymptomatic, meaning you guys will get infected and you won't know that you have it, and you can spread it to others because you don't have any symptoms of disease. And that's really quite high. In fact, that's how COVID-19 got into this country. If you remember, Trump put up, I don't know what you want to call it, restrictions on people entering the country. And I think with China, he said nobody from China is going to enter the country. Okay. And then from other countries, they had to go through quarantine or something like that. But it came into the country because of asymptomatic people bringing it into the country. After 2 to 14 days, the patient, if they have symptoms, they'll get symptoms. But if they're asymptomatic, they'll never get symptoms. The estimated death rate ranges from 0.1% to 3%. This is a bit old now. It's probably 6 months out of date. The actual death rate in the United States was 1.2% of patients who became infected died. And as I stated, the death rate will vary depending on the variant of COVID-19. The Delta strain had the highest mortality rate. Prevention, use correct hand washing. Wash your hand for at least 20 to 30 seconds. Keep your distance from other people, especially if they're coughing. Don't touch your face with your hands. If you're sick, masks are recommended to keep the COVID-19 from spreading from you. It's best to stay home and cough into your elbow or tissue. Any question about any of that? All right. Any questions about COVID-19 in general? If not, let's talk a little bit about viruses and cancer. Some human cancers are caused by viruses, and it's about 10 to 15% of human cancers are caused by viruses. Some examples, human papillomavirus, like cervical and skin cancer, is caused by a virus. Hepatitis B virus and hepatitis C virus are known to cause liver cancer. However, not all cancers are caused by viruses. Like human breast cancer and glioblastoma, there are no examples that we know of that these cancers are caused by a virus. So they're certainly just not associated with a virus. This is telling you a little bit about cancer. I'm not going to quiz you on that, so I'm going to skip over it. There's two main ways that viruses can cause cancer. One is the virus infects the cell, and then the virus integrates into the host chromosome, forming a provirus. This is a permanent genetic change in a cell, or at least in a eukaryotic cell. So that can cause mutation when a virus enters the DNA of a patient, and mutation will increase the risk of cancer. You know that because exposure to X-rays and other forms of radiation increase the risk of cancer. All right, that's one way that viruses can cause cancer, and that is they're integrating into our DNA and changing the DNA. And it's true they can mutate the gene they integrate into, but there's other things they can do to muck up the DNA, like they can bring in a promoter so they can turn on a gene which is normally turned off, and they can turn off a tumor suppressor gene, and there's other things they can do. But the point is they're integrating into our DNA, and that causes problems for our DNA. The second way a virus can cause cancer, if it is an oncogenic virus, this virus carries an oncogene which causes cancer. An oncogene is a mutated form of a normal gene involved in cellular growth. And if the cell gets this mutated gene that is involved in cellular growth, the cell will just grow uncontrollably. So the oncogene, when it infects a cell, it causes the cell to grow uncontrollably. And that increases the risk of cancer. So mutated oncogenes, which is all oncogenes, help transform normal cells into cancer. So an oncogene is carried by some viruses, and we call these oncogenic viruses. Sometimes the oncogenic virus can even integrate into the host cell DNA, and then the cell has two risks of getting cancer. The oncogene increases the risk of cancer, and then the provirus increases the risk of cancer. Any questions about how viruses increase the risk of cancer? The virus that is most likely to cause cancer in humans is human papilloma virus, causing cervical and skin cancer. Okay. Any question about any of that? All right. We talked about in animal cells that can get viruses which are lytic, follow the lytic life cycle, and then viruses that can follow the budding life cycle. There is another life cycle, and I did mention there's the persistent viral infection, which kind of follows the budding virus, except it's not an envelope virus. It doesn't bud out of a membrane. It is a naked virus, and it's shed by the cell in a vesicle. You don't really need to know about the persistent viral infections, but it's another life cycle of animal viruses, and it's very similar to budding. It's just the virus doesn't obtain an envelope from budding out of the cell. It forms a vesicle and then comes out of the cell. All right. There's another virus life cycle we need to talk about, and it is the latent viral life cycle. The latent viral life cycle usually happens in a budding virus. When the virus is actively multiplying, it's in the budding life cycle. But the virus can go latent, where the virus remains in an asymptomatic state, perhaps for long periods of time. When the virus is in the latent life cycle, it does not cause disease. And the virus is just sitting out, being more or less dormant. And then the virus can come out of latency and reestablish the budding life cycle. Some examples of that are cold sores caused by herpes simplex virus 1. When you have the cold sore, you're in the budding life cycle and you have symptoms, the cold sore, of a viral infection. And then when the cold sore goes away, the budding life cycle stops, the virus stops multiplying and really goes pretty dormant. And then the virus goes into the latent life cycle. Another example is chickenpox virus. When the child is first infected, he comes down with chickenpox and the virus is in the budding life cycle. While the child has symptoms of chickenpox, the disease of chickenpox, including the pox marks, the virus is in the budding life cycle. The virus can then go latent, and this latency can last for decades. Usually it's a child who gets chickenpox, and then the virus comes out of latency and causes shingles. So the first time the child is infected or the person is infected, they get chickenpox. All other times the virus comes out of latency, the patient will get shingles. And when the patient has shingles, the virus is in the budding life cycle. The triggers for the virus to switch from latency to the budding life cycle and then cause disease are usually a stressor. For a cold sore, the stress is usually coming down with a human cold. For shingles, the stress is usually the patient becomes weakened in their immune system. And this usually happens when the patient becomes elderly. Why the virus hits a child most of the time and then goes latent until the patient is elderly and then comes out as shingles. Any question about any of that? All right, there are some persistent viral infections. Disease of a persistent viral infection process occurs over a long period of time. The detectable levels of the virus build up over a long period of time. And the virus is never usually cleared by the host immune system. The persistent viral infection may be fatal. For example, in the first, 10 years of HIV and AIDS, it was a persistent viral infection. And the HIV virus in the patient would increase with time and then would blossom out. And the patient normally died. So it would be fatal. Okay. But the persistent viral infection does not have to be fatal. Sometimes the infection can be tolerated. Like when patients in the United States become infected with cytomegalovirus, these patients will tolerate the virus and then they'll have cytomegalovirus for decades, perhaps for their lifetime. And the host will tolerate it, especially if the virus isn't replicating at high numbers. It's easy to tolerate. There are a few cases in the United States of varicella zoster or chickenpox where the patient never gets better. They always have symptoms, usually of shingles. And this is probably less than 1 in 1,000 patients. The shingles never go away. What happens is the virus is always in the budding life cycle and the shingles are just tolerated. Some other herpes viruses follow the persistent viral infection and they're tolerated. An example of a fatal virus, I'm sure you've never heard of, subacute sclerosing panencephalitis. It's actually caused by the same virus as measles virus, but when you get subacute sclerosing panencephalitis, it just gets worse and worse. And here you see the green bar showing the persistent viral infection. It starts out low numbers, and over many months or years, the numbers just build up until there becomes a bloom. And as you can see, the graph is broken. It's because oftentimes the patient dies. So the patient never gets better. It's different from an acute infection where initially the patient becomes infected. We have a latent period where the patient is replicating the viruses, and then the virus replicates and we have signs and symptoms. And we have the worst signs and symptoms when the patient is ill, and then very quickly the patient gets better. This could be an example of the human cold. Very quickly, the virus infects the patient. The virus numbers quickly increases. The patient has the highest incidence when the patient is sick, and then the virus quickly decreases, and then it's gone. Okay, an acute infection, very quick, very high numbers of viruses while the patient is sick, and then it's very quick and it goes. It's different from a latent infection, which if you have chicken pox, for example, initially the child will get infected. The number of viruses being shed will be very high. The patient will be at their sickest right here. Then the patient will get better. And then the disease goes away. The virus goes latent. And that could be for months or years. And then the latent infection occurs. The patient gets shingles. And most of the time, the viral load is nowhere near it was with the acute infection. And then the patient gets better and the virus load decreases to zero, a latent infection. So that's what happens most of the time when patients get shingles. And before that, it's called chickenpox. Very different from a persistent infection, which this one just continues and it never ends. Even if it's tolerated, you bring this out to the end here. Okay, or it just keeps climbing like that. Any question about any of these? All right, if not, are there any questions about viruses and virus disease? All right, if not, let's move on to prions. Prions are a proteinaceous infectious particle. They are infectious proteins. And they can be inherited where a patient gets a gene which is more likely to become a prion, and that gene obviously codes for a protein. Most prion diseases are transmissible by ingestion or by transplanting an organ containing the prions or by surgical instruments. Prions are very difficult to kill. And in surgery, if a patient has prions, the surgical instruments will get the prions on the instruments. And then when the instrument is sterilized, it does not destroy the prion. To destroy prions, you need something like 1,000 degrees temperature to destroy the protein. And they don't really know why prion proteins are so difficult to destroy. Like they planted prions in the soil, and then they looked to see how quickly the prions disintegrated. Because normally proteins, if you put them into the soil, they will be digested by other organisms and the protein will go away. Well, they looked at the prions in the soil after a year being in the soil, and the prions were still quite high in the soil. I don't know what the decrease was, but it wasn't like 90% of the prions went away. The prions, the majority of the prions were still there even a year after being put into the soil. With organ transplant, they do check a patient and the transplanted organs. So nowadays, this is very unlikely to happen where a patient is given an organ transplant and then they get the prion. The same thing with surgical instruments. They usually test the patient before surgery. And if they have prions, they know that something dramatic has to happen to those instruments. There is a way to treat the instrument because normally 1,000 degrees Fahrenheit would destroy the surgical instrument. But you can treat the prions with proteinases. These are enzymes that digest proteins, and then that gets rid of prions. And that's one way you can treat surgical instruments if you're operating on a prion infected patient. The other thing is you just throw away the instruments. Prions cause spongiform encephalopathy. These are literally vacuoles in the brain, making the brain look like a sponge. Let me show you a picture of that. Here we go. These three are brains of prion infected brains, and there's a normal brain. All right. And if you look at that one and that one in a little bit less degree, this one, it kind of looks like a sponge, giving the term spongiform. In animals, the most common prion is and was bovine spongiform encephalopathy, which is more commonly called mad cow disease. However, it has been seen before. There's a disease in sheep called scrapies that sheep farmers in Europe have been dealing with for centuries. Okay. And we'll talk a little bit more about that. It's possible that scrapies was how mad cow disease came about. We'll talk a little bit about that. And then in animals, it's usually called something like cervid chronic wasting disease. On humans, we don't call it, obviously, bovine spongiform encephalopathy. We don't even call it that Homo sapien spongiform encephalopathy. We usually call it Creutzfeldt-Jakob disease, or variant Creutzfeldt-Jakob disease, or Gerstmann-Strauss-Lasser Schinker syndrome, named after the people who first discovered it in families, and fatal familial insomnia, and then Kuru. So these are the terms we use for prion infected disease. With these two, it's usually certain families. They have a gene that is more likely to go prion, and the same with this one. All right, I already talked about that. I should move that slide up since I talked about it. I think I went past something. Oh, here we go. Prion, proteinaceous infectious particles, cause Kuru. Kuru was a disease seen in the Forb tribe in Papua New Guinea. Let me see if I can blow this up. The Forb tribe is this red region of Papua New Guinea right here. The disease caused patients to be unable to walk, loss of the ability to swallow or chew, drastic weight loss, and then led to death. How it was transmitted? The Forb tribe had an unusual ritual, and it was a funeral ritual. What the Forb tribe did was when one of their relatives died, they would cook the person and then eat their dead relative. Okay. And apparently they did this for a while, and somebody came down with a prion disease, and they cooked the person's brain and then ate the brain, and then the prion disease continued in the Forb tribe. Let's see if I've got another page on the Forb tribe. No, I don't. So I'll mention that women noticed that when they were eating their dead relatives, that they and their family members would come down with Kuru. And so women in the Forb tribe stopped feeding their children and themselves their dead relatives. So the cannibalism stopped among the women and the children, but the men continued the funeral ritual. And then the white Europeans came to the Forb tribe in Papua New Guinea, and the missionaries said, "Oh my gosh, they're eating other people. They're practicing cannibalism. We got to stop this." And the missionaries actually did something that was good, at least in my opinion, and that is they stamped out cannibalism. They passed a law, and I think they imprisoned people who practiced the funeral ritual. And that got rid of Kuru in the Forb tribe. And that happened like in the 1920s and 30s. The missionaries were just somewhat intolerant, and they could not stand seeing the Forb tribe practicing cannibalism. And it wasn't normal cannibalism. The patient died on their own, and it was part of the funeral ritual. And then they ate them as part of the funeral ritual. Okay. Anyways, Kuru is now pretty much gone from the Forb tribe, largely because of the discontinued practice of they no longer carry on cannibalism. All right, I showed you these. Kuru, Scrapie's infected brain. I don't know if that's a human brain, but that's a human brain, and that would be a human brain. That might be a brain of sheep, because that would be the animal that gets Scrapie's. It is true that occasionally Scrapie's would transfer from the sheep to the sheep farmer. Like I said, Europeans have been dealing with Scrapie's, a disease of sheep, for centuries. And it didn't transfer very readily to the sheep farmer. But every once in a while it did. So maybe that is a Scrapie's and a human brain. All right, how does a prion replicate and cause disease? Remember, a prion is a proteinaceous infectious particle. The way that a prion causes disease is there's two forms of a gene. PRP superscript C is the normal gene. and it is encoded by a cell. It makes a cell surface protein required by a nerve cell. And then there's the abnormal form of the, I don't want to call it gene, which causes the PRPS superscript SC, or standing for Scrapes form of the prion. And this is the prion protein. The PRP superscript C and the PRP superscript SC are the same protein. They have the same sequence of amino acids. The difference is just the folding pattern. So PRP C has the normal folding pattern, and then PRP SC has the prion folding pattern of the protein. The PRP SC protein comes up to the PRPC protein and causes the normal protein to take on the folding pattern of the prion protein. Now the cell has two prion proteins in it. The cell says, oh gosh, I need more PRPC protein. And so the cell makes more PRPC protein. The trouble is, this cell has two prion proteins in it. And if one of them comes up to the PRPC protein, it converts it into the Scrapes protein. So more and more Scrapes protein is made in the cell. And as more and more of the PRPC protein is lost, the cell makes more of the PRPC protein. So abnormal Scrapes protein accumulates, especially in the brain cell, and that causes the plaques to form in the brain. The proteins tend to stick together, and this eventually causes cell death of the nerve cell. This is very bad if it's in the brain cell. Okay, or in the brain, sorry, if it's a cell in the brain. Anyways, that's how the Scrapes protein replicates itself. It comes up to the normal protein and converts it into the prion form of the protein. Mad cow disease was the most well-known example of prion disease, where in Britain, they had the atrocious habit of grinding up cow brains and putting it on grain that they fed to cows. And what happened was there was a brain that had prion disease. Possibly the first case was Scrapes, meaning they put sheep brains, ground up sheep brains also, and then put it on the grain, and then fed the grain to the cows. And then the cows would eat it and come down with prion disease, causing mad cow disease. This caused an epidemic of mad cow disease in Britain, and in British people and British cats, we're talking about domestic kitty cats and zoological animal zoo cats, came down with prion disease. There's been about 150 confirmed cases in England of people getting prion disease from eating prion tainted beef, and then about 150 cases of British cats coming down with prion disease from eating prion tainted beef. Surprisingly, dogs were resistant to getting prion disease, and they were eating about the same food as British cats. So dogs must have been much more resistant to getting prion disease than cats. And I guess much more resistant than people too. Any question about prion disease?
I did have a question.
Uh-huh.
Is the mechanism of this disease, so like the conversion of the normal protein, is this common for all other prions, or is that just this one specifically? Or is it like similar?
It's commonly known for all known prion diseases. And that if you think about it, how would a protein replicate? Because it has no DNA. If it gets into a cell, how is a protein going to replicate? It has to have some way of replicating to cause disease. So probably it happens to all prion diseases. It's just that we haven't studied all prion diseases. They've studied mad cow disease and they've studied scabies. I don't think this has actually been studied for any other case of prions. Okay.
Gotcha.
But
Thank you.
they think it happens for all of them because the protein has to multiply to cause the prion disease.
Gotcha.
Okay,
Thank you.
sure. Good question, though. Anything else I want to say about prion disease? The United States got very few mad cow disease cases because we did not import British beef or British grain, or did we buy British cattle? A few European cases occurred because, mostly among cows, because they imported British cows, or like Canada, they imported British grain, and that was fed to cows, and then it resulted in some prion diseases in Canada. But the United States slapped up a quarantine, and so we weren't importing British grain or British cattle. And so we were very few cases of prion disease. However, prion disease in cows does happen normally just by mutation. And I never mentioned that, but there have been about 5 cases of prion disease known in cattle in the United States. And each of these cases, well, 3 of the cases happened by mutation, and that is the normal gene mutated into a form that was much more likely to generate a scrapese protein. And I don't want to say it's a scrapese protein directly because it's still a normal protein, but it's a gene that's much more likely to fold into the scrapese protein. Okay, and that's what happens in the, where do I have that? Creutzfeldt-Jakob disease patient. These patients have a gene which they're fine until about middle age, and then the protein, one of them converts into the prion form of the protein, and then the prion form of the protein converts the normal protein, which is mutated because they're in the Creutzfeldt-Jakob family, but it changes that protein into the prion form. And that's how they get prion disease. In these families, almost never do the patients come down with prion symptoms before the age of reproduction. So they're perfectly normal in childhood and adolescence and young adulthood. It's not until they reach middle age that some of them start showing signs of prion disease. Any question about any of that? My point is that there are variants of genes which are more likely to change into the prion protein. And this happens by mutation all the time. And 3 of the cattle seen in the United States, that happened by mutation. The parents of this cattle apparently just mutated, and then the cattle had the mutation, and then the cattle mutated, and the prion protein was made, and then the cattle got prion disease. Any question about that? The other 2 cases were, I think, from cattle imported from Canada. And Canada didn't have many cattle that were infected with mad cow disease, but they did import British grain. And so a few Canadian cattle came down with mad cow disease. And the United States did import cattle from Canada. And so 2 of the cattle we got that later came down with mad cow disease were obtained from Canada. All right, any questions about scrapies? If not, let's talk about viruses and viroids. Plant viruses are not a topic that we're going to talk about much because they do not infect animal cells. They do not cause disease among humans. They obviously cause disease among plants, but they don't cause disease among humans. So we're not going to talk much about plant viruses. But plant viruses resemble animal viruses in many ways. They do differ in that plant cells have a cell wall and animal cells do not. So the plant viruses have to come through or have some mechanism of coming through the cell wall. Plant viruses do cause many diseases of economic importance for economically important crops, like the viruses, plant viruses can cause disease in beans, corns, sugarcane, potatoes. The only one I know is tobacco mosaic virus can cause wilting or spots in let's see, potatoes and tomatoes, as well as tobacco. Okay. Because of the tough cell wall, plant viruses usually enter the plant through wounds or via a parasite. And that parasite can be a nematode, a fungus, or an insect. So the parasite damages the plant, and then the virus can get in. The infected plants can spread the virus in pollen or seeds. And obviously, an insect can eat the infected plant and then move on to another plant, spreading disease that way too. Any question about plant viruses? The only plant virus I'm going to mention is, well, I already mentioned tobacco mosaic virus. There's another one, spindle tuber formation of potatoes. It causes the potato plant to make smaller potatoes than normal. So if you have a normal potato about that size, the potatoes will be smaller. If it has the spindle tuber virus. Viroids are like a naked RNA virus. So a naked RNA virus would have a protein coat surrounding an RNA molecule. Well, a viroid, all it is a naked RNA molecule. So think of it as a naked virus without the protein coat. This naked RNA molecule has nothing other than the RNA. There's no protein coat. It is seen only in plant pathogens. Oh, I got that wrong. The potato spindler tuber viroid is a viroid, not a virus. And I need a little picture of that. If the plant gets the viroid, potato spindle tuber viroid, instead of getting normal big potatoes, they'll get little potatoes. There are no viroids known in animals. So there's no viroids that we know of that infect animal cells. Any question about any of that? All right. My last slide is recently they found some huge viruses. Megavirus used to be the largest known virus, a little less than 500 nanometers. I think it was about 440 nanometers. Well, now they found Pandora virus, which is, well, about the diameter would be about 700 nanometers. It's 1,000 nanometers long and 500 nanometers large. This is actually bigger, the Pandora virus. It's bigger than some prokaryotic cells. The smallest prokaryotic cell is about the same size as the megavirus. Both of these two very large viruses infect amoeba. Any question about any of that? Any question about viruses, prions, or viroids in general? If not, that's it for this lesson. Let's go to the next lesson. I guess I should save that. See how slow this is? Oh, I need to open that up. Chapter 14. Principles of disease and epidemiology. This slide is showing you the major goals of this lesson and a rough outline. Know the terms. I've only got 3 here: pathology, etiology, and microbial antagonism. But with this lesson, if you know the terms or learn the terms for this lesson, you'll actually learn about half of the material for this chapter. Meaning this term, this chapter is very heavy on terms, and it's an important part for the principles of disease and epidemiology. Understand the relationship of the normal microbiota and its host. Understand the stages of disease development, meaning when a patient becomes infected, the disease normally goes through different stages as the disease develops. And then lastly, understand about nosocomial infections and how to control them. Any questions about what we're doing? All right. Pathology, infection, and disease is introducing 3 terms to you. Pathology is the study of disease. Etiology is the study of the cause of a disease. For example, the etiological agent of anthrax is Bacillus anthracis. And this was the first microorganism that was shown to cause disease. Actually, it was the first organism, period, that was shown to cause disease. Pathogenesis is the development of a disease. Infection is the colonization of the body by pathogens, or it may be the presence of a microbe where the microbes are not normally found. For example, I normally have Staphylococcus epidermidis all over my skin. And while it's on my skin, it does not cause an infection. However, if it were growing in the body at some other location, such as in my lungs or beneath the surface of the skin, that would cause an infection. Any question about any of that? Disease is an abnormal state in which the body is not functioning normally. So when you get an infection and a microbe grows in you, it causes your body to not function normally. And then you have signs and symptoms of disease. Now, when we're talking about microorganisms, it's important to know that few microorganisms are pathogenic. Very few of them cause human disease. Some of them actually can benefit the host. And we need these microbes to help us. And these microbes that I'm talking about that benefit the host are the microbes of the normal microbiota. The normal microbiota forms a relationship with its host. The normal microbiota is a large community of microbes found in association with its host. And this can be all animals and plants. We have a large community of microbes living with them. These communities are known as the normal microbiota. So you could say the human normal microbiota, or you could say the domestic kitty cat normal microbiota. The microbes that normally permanently colonize the host. And when a member of the normal microbiota colonizes the host, it usually lives with the host for the lifetime of the host. So for a very long time at the very least, and normally the lifetime. When you're born, you have very few normal microbiota. Okay, you may have a few normal microbiota, which mainly you pick up during birth, but then after you're exposed to the real world, the real environment, you get your normal microbiota. Many different organisms are given to you largely from your parents. The normal microbiota normally permanently colonize the host. They're only found in certain regions of the body. And it's seen that disruption of the normal microbiota throws the host balance out of whack. And the normal microbiota host balance, if it's thrown off balance, it's associated with disease and impaired immune function. For example, when a patient is given antibiotics, that hits the normal microbiota, and the patient frequently will have signs and symptoms of either diarrhea or constipation. And most of the time, that's short term, and then the patient returns back to normal. Any question about any of that? The return to normal may be because of the establishment or reestablishment of the normal microbiota. But it could be that the host is also just adapting. The human microbiota is normally found on the skin and the surface of the body. It's normally found on the mucosa open to the body, like the nose and the throat, the mouth, the eyes, the intestines, or actually the entire gastrointestinal tract, and then the lower urinary and reproductive systems, like the lower urethra on both men and women, is colonized by the normal microbiota. The higher urinary and reproductive systems are not normally colonized by the normal microbiota. Like the kidneys does not have a normal microbiota. Okay. Examples of places where the microbes are not found include all internal organs and tissues, like the blood, the cerebrospinal fluid, the muscle, the nervous tissue, and the bone. As long as the tissue isn't open to the outside, there's normally not normal microbiota. Okay. And we can talk about the large intestine. It's usually connected to either it's connected to the mouth and the anus. And so it gets bacteria mostly from the mouth. Any question about any of that? All right, the normal microbiota differs from the transient microbiota. The transient microbiota may be present for days, weeks, or months, but unlike the normal microbiota, the transient microbiota comes and then goes. It doesn't stay around. An example of a transient microbiota that I once had was I went and visited my aunt, and she had a Staphylococcus aureus infection on her skin. And when I went to see her, she insisted on hugging me. Okay. And I didn't think much about it. And she was receiving antibiotics for her skin infection. So I didn't think it would be a risk. But I sampled myself after that. And back then, I used to sample myself every term. I would actually encourage my students to do it too, and sample their skin to see if they had Staphylococcus aureus growing on their skin, because about 20% of Americans have Staphylococcus aureus growing on their skin. And this is a human pathogen. These patients are more likely to come down with the skin lesion. Okay. Anyways, for years before that, I had no Staphylococcus aureus after my skin. Then after hugging my aunt, I had Staphylococcus aureus on my skin. And I said, geez, that's not good. Must have picked it up from Aunt Larry. Fortunately, the next time I sampled my skin, the Staphylococcus aureus had gone away. It was no longer on my skin. And so it was a transient microbiota for me. Any question about any of that? If no questions, let me state that the normal microbiota establishes a relationship with their host. Humans have about 1 × 10⁻13 human body cells, but humans have about 1 × 10⁻14 bacterial cells, meaning there are 10 times more bacteria in and on you than you have human cells. And that begs to ask the question, are we more microbe than we are human? And don't let that freak you out too much. Okay? Because yes, you have more microbes than you have human cell, but you're still human. And everybody has a normal microbiota. So that's part of the normal human too. But yeah, you are more microbe than you are human, at least in cell numbers. Here are some normal microbiota. Let's see if I can blow this up. Bacteria in the nasal epithelium. We're seeing cocci, which are the bacteria normally seen. So it's part of the normal mucosa, normal microbiota. Bacteria on the lining of the stomach. So that's the stomach. Here's the bacteria. That is not bacteria. That is, what is that called? Structures of the cells of the stomach. And then a bunch of normal bacteria in the large intestine. There we have a cocci. There a coccobacillus. There a bacillus. There's another coccobacillus. That looks like food particles. I'm not sure what those are. That might be, no, that's too small to be a human cell. Too large, too small to be a human cell. Any question about any of this? All right. Recently, in the last 10 or 15 years, we've discovered that the humans have a microbiota in regions we never thought they did. Like they've sampled the adult female bladder and found that the adult female bladder has a normal microbiota. So there are bacteria growing in the adult female bladder. Curious to find out what the cells would be eating, because I don't know what they'd be eating. I doubt they'd be digesting urea, but that's certainly a possibility. There are normal microbiota inside the human breast tissue. So mother's milk is not sterile. She has bacteria in her breast, and that bacteria goes into the mother's milk. And then when the infant consumes the mother's milk, they will get the mother's normal microbiota that's in the mother's milk. Any question about any of that? All right, they haven't looked at whether males have a normal microbiota in their breast tissue, but the male breast is very different than the female breast. They haven't looked to see if males have any normal microbiota in their bladder as well. But you have to remember the urethra is much longer in males than it is in females. So it's not terribly surprising that females will have a normal microbiota in their bladder and males may not, just because the urethra is so much longer. This is one which I'm not sure why it came as a surprise, but the human placenta has its own unique microbiota, cells that specifically grow in the placenta. And if you think about it, a female doesn't usually have a placenta. It's only when she is carrying a baby. Although she has a primitive placenta. Think about that. Yeah, she has a primitive placenta when she's shortly after she's ovulating or right before she's ovulating, something like that. Yeah, that primitive placenta then disintegrates if the female does not become pregnant. So the placenta is mainly there when she's carrying the unborn baby. All right. I mentioned that the normal microbiota forms a relationship with its host. And this relationship is either microbial, oh, what's the term? Sorry, lost it. It's microbial mutualism or commensalism. Okay, the normal microbiota does not normally cause disease, and in fact, it's essential for the health of the host. The normal microbiota can engage in microbial antagonism with a bacteria that causes disease. Microbial antagonism is competition between two different microbes, such as the normal microbiota preventing the growth of pathogens. The normal microbiota may occupy the niche, like I have lots of Staphylococcus epidermidis on my skin and a few other bacteria as well. And if a human pathogen were to come down, it may not have room to occupy my skin. So the normal microbiota may occupy a niche that the pathogen might need to get established. The normal microbiota can compete for resources, like a Staphylococcus epidermidis on my skin consumes CBM. And if a human pathogen were to consume CBM, that would be direct competition between the human pathogen and the normal microbiota. The normal microbiota can produce acid, making the environment inhospitable to the human pathogen. For example, on our skin, the human normal microbiota make the skin more acidic, and they lower the pH of the skin, the normal microbiota, by about 1. So the normal pH is around 5.5, assuming you haven't had a shower recently or a bath recently. Part of that lowering of the pH is by the protein that is made by the bacteria. Any questions about any of that? All right, the last way that a microbe can antagonize its host, not its host, antagonize a human pathogen is the bacteria can produce a bacteriocin. A bacteriocin is like a very narrow-acting antibiotic. A bacteriocin does not inhibit all bacteria. In fact, it only inhibits bacteria that are relatively similar to the cell that made the bacteriocin. For example, E. coli can produce a bacteriocin, and this bacteriocin is a protein that inhibits the growth of Salmonella or Shigella. So if Salmonella were to colonize your gut and you had E. coli making a bacteriocin, the Salmonella may not get established because of the bacteriocin. I often wondered why people weren't using bacteriocins as an antibiotic, because as I stated, a bacteriocin inhibits or stops the growth of bacteria. Not all bacteria though, because it's very narrow in its mode of action. And it's much more narrow than a drug that only hits gram negatives, for example. Like polymyxin B is a drug that only hits, do I have that reverse? Yeah, it's a drug that mainly hits gram negatives. And the story is that bacteriocins are now being experimentally used as a new antibiotic. And if they do develop bacteriocins, and it's shown that it's a good choice for an antibiotic, it would be good to give the patient a bacteriocin instead of an antibiotic, because the bacteriocin is much more narrow in its range of action. So if you give the bacteriocin that inhibits Salmonella and Shigella, you'll inhibit those two species, but you will not inhibit other species that are in your gut or on your skin. Any question about any of that? All right. Probiotics are the consumption of live microbes to exert a beneficial effect. Like all live cultures of yogurt have a live bacteria in them, which can then become a normal microbiota growing on your skin. This bacteria in yogurt is good to have because it reduces the risk of a bladder infection or a UTI and a few other diseases. Let me see if I got that stated. I do not. So if you give the patient a bacteriocin, it will not inhibit most of your normal microbiota in your intestines. All of the bacteria that are not related to Shigella will not be affected. The bacteria that are related to Salmonella and Shigella would be inhibited by the bacteriocin. But something that's really different, like Staphylococcus aureus, would not be inhibited by the bacteriocin. Any question about any of that? All right, I think I already mentioned this. Your normal microbiota lives in symbiosis with the host. Symbiosis is when two different organisms or populations live together. The normal microbiota and the host are in symbiosis, and specifically, they're in one of two forms of symbiosis. They can be commensalism, and that's a commensalistic symbiosis with the host, or they can be mutualism, where the normal microbiota gets a benefit, such as its home. It may get food from its host. The host gets a benefit out of it either, meaning with E. coli, it helps us digest our food. So E. coli in our intestines is a mutualistic symbiotic, forms a mutualistic symbiotic relationship with us. Commensalism is a little harder to come up with one that is strictly a commensalist, because almost all of the microbes that are part of our normal microbiota, it can engage with microbial antagonism, occupying the niche the pathogen may occupy, or competing for the resources the pathogen may need. So it's hard to find one that's exactly a commensalistic organism. But something that's close would be Micrococcus luteus on my skin. It gets a home, the Micrococcus luteus. It gets some nutrients like sebum on my skin, giving the bacteria food and energy and carbon source as well as a home. That differs from symbiosis, where one organism benefits, but it benefits at the expense of the other. And that is a parasitic relationship, such as a tick living on you or your cat. The tick benefits because it gets a home, it gets nutrient from your cat by biting the cat, sucking the cat's blood. The cat doesn't get any benefit out of it. In fact, because the cat is being bitten, the cat can be at increased risk of getting disease from the tick or flea. And so that's a parasitic relationship. Any question about any of that? All right. Let me state that when you give an example of a mutualistic or commensalistic bacteria, that you need to remember that it's really hard to find one that's strictly commensalistic. Most of our normal microbiota will at least prevent the growth of a pathogen just by occupying the niche. Like Staphylococcus epidermidis is all over my skin, and so there may not be room for Staphylococcus aureus to fall on my skin and then begin growing. Any question about any of that? The normal microbiota does not normally cause disease. However, the normal microbiota may cause disease under some circumstances. If the normal microbiota does cause disease, we call it an opportunistic microorganism, where some normal microbiota becomes opportunistic. And that could happen like when the Staphylococcus epidermidis is growing where it should not be growing, like internally instead of on the skin. Staphylococcus epidermidis normally does not cause disease in healthy individuals, but it may cause disease in an AIDS patient. The AIDS patient has no immune system, and so Staphylococcus epidermidis may bloom with the loss of the immune system and then cause disease. This is true of all patients with a compromised immune system. They often can become infected with an opportunistic pathogen. Some examples: E. coli is normally found in the gut. Staphylococcus aureus is normally found in the nose and the throat. If it's growing anywhere else, we would call it a pathogen, such as E. coli growing in the lungs. That would be an opportunistic microorganism. Staphylococcus aureus, once again, growing in the lungs, would be an opportunistic microorganism. Any question about any of that? All right, this is the third and final time we'll talk about Koch's postulates. Koch's postulate is the tool that we use to show that a given organism, microorganism or some other organism, causes a given infectious disease. Remember, Robert Koch first proved that Bacillus anthracis causes the disease anthrax. And this is the first time that Koch's postulates were used as well as the first time a microorganism was shown to cause disease. Any question about any of that? All right, let's go through Koch's postulates. Oops. With Koch's postulates, we have a sick bunny or sick individual. To have Koch's postulates occur, you have to obtain the cells from the sick individual and then get them to grow in vitro, meaning in a laboratory, and you need to get them to grow and then purify it so you get a pure culture of that organism that you think may be causing disease. You then grow up that organism and inject it into a healthy bunny or a healthy individual, meaning it does not have the signs and symptoms of disease. If that bunny then becomes sick, why it's shown on its side, oh, the poor bunny, he's sick. That highly means that this organism causes that disease. There's a few other things you need to do. You have to be able to re-isolate that organism causing the disease from this bunny. And then you have to show that this disease is similar to this disease. And then lastly, the organism must be present in every case of the disease. Like in AIDS, we know that HIV is present in every case of a patient who had AIDS that we know of. Okay? Any question about any of that? All right. Wait a minute, I went the wrong way, I think. There are exceptions to Koch's postulates. What happens if you have a microbe with a unique culture requirement and you cannot get it to grow in vitro? Two examples are the bacteria that causes syphilis, something palindrome, and then the bacteria that causes cat scratch fever. Is that Bartonella henselae? These two bacteria cannot grow in vitro. We cannot get them to grow in the lab. And people have tried for over 100 of years to get these microorganisms to grow. They have a unique culture requirement, and we don't know what it is, but that additive has to be added before the microbe can grow. And we don't know what it is, so we haven't figured out what we can add to get these organisms to grow in vitro. So if these organisms can't grow in vitro, we can never grow it in vitro and then inject it into a healthy individual and then watch that healthy individual come down with disease. So that's an exception to Koch's postulates. What happens if multiple organisms cause the same disease, signs, and symptoms? Then you can't use Koch's postulates to show that an organism causes one disease. For example, there's many different viruses that, and we're talking about different viral families, that can cause the human cold. So if you were to isolate the cold virus from one sick individual and then add it to another healthy individual, even if that individual comes down with the cold, that doesn't prove that that virus caused the cold, because multiple viruses can cause the human cold. What do you do if some species can cause different disease conditions? Meaning one organism can cause more than one disease. Like salmonella can cause both typhoid fever and salmonellosis. And then what if you do if there are moral and ethical reasons for not infecting a human patient with the disease-causing organism? Like HIV, we've never infected it, grown up HIV virus and infected it into a healthy individual to see if they then later develop AIDS, because that would be unethical. That would be essentially, for the person getting AIDS, that would be a death sentence. So what do we do if we can't use Koch's postulates to show that a given microorganism causes a given disease? Anybody have a guess? Nobody has a guess? What we do is we do vast epidemiological studies. And with HIV, we can't grow up HIV in vitro and then inject it into a patient and then later see if the patient becomes infected, not infected, later comes down with AIDS. We can't do that for ethical and moral reasons, but we can study vast number of people and follow them over time in an epidemiological study. Some of those people will be HIV negative and then will become HIV positive by picking up the virus on their own. And then we can study those patients and watch them later become AIDS patients. And that's how we know that HIV causes AIDS from the epidemiological studies. Any question about any of that?
I just wanted to remind you that we end at 6:50.
Okay, yeah, I noticed that. Let me talk about this slide and then we'll end. Classifying infectious diseases. Diseases can cause certain symptoms. A symptom is a change in a body function that is experienced by a person as a result of a disease, but nobody else can observe that symptom. So it's subjective. The symptom is not apparent to another observer. Like when a patient gets a headache, nobody else can confirm that that patient has a headache because there's nothing for us to measure or to observe to verify that the patient has a headache. A sign, on the other hand, is a change in the body that can be measured or observed as a result of disease. Like a temperature of 102, we can measure that. And so that is a sign. And these are the textbook definitions for what a sign and a symptom are. Now I'm going to warn you that when you go out into the clinic, if you're not in the clinic already, they will use the terms signs and symptoms interchangeably. And that's the way the common usage of these two terms are. So the textbook definition is a symptom is one that you can't measure, and a sign is a symptom that you can measure. A sign is a sign, I guess I should say, that you can measure. Okay, any question about that? Now, in this class, I will generally use the terms sign and symptom interchangeably because that's the way most clinicians are, meaning they will use them interchangeably too. A syndrome is a specific group of signs or symptoms that characterize a disease. Like AIDS patients typically have certain signs and symptoms characteristic of their disease. Like their immune system becomes weak and then will go away, and they will have certain signs and symptoms, like they'll have increased risk of opportunistic infection. I'm trying to think of what it is, like they'll have a increased risk of getting a hairy tongue. All right, any question about any of this? If not, I'm going to stop here. And we'll continue here in our next lesson. Remember, I'll be in the lab only from 7 to 7:15. If you have questions, come to the lab during that time and I'll answer your questions. If no one is there after 7:15, I will log out. All right, I'll see you.
Thank you.
Bye.